Today I'm going to discuss about mRNA molecule and also we will see the two inherent features of mRNA, that means the reading frame and also the genetic code. As all of you may know that mRNA is a product of transcription. So by transcription we will get the mRNA, and these mRNA molecules will be subjected for translation to produce a meaningful protein. So we will see how this informations from the DNA is decoded into the mRNA, or in which format these informations are stored in the mRNA molecule. That is the first question. The second thing is how this molecule is being utilized as a template for translation. So for all those things, or in short, for a better understanding of translation, we need to know about mRNA molecule and their features. This will give you a basic idea about mRNA molecule. This may be very basic, and all of you may know this one. These are, or mRNA molecules are the a nucleotide sequence, and these sequences will be decoded, or they are acting, these nucleotide sequence within the mRNA will act as what we call the codons, and these codons will determine the amino acids for protein synthesis. And one important or interesting feature is that most of the mRNA molecules will be having a very short lifespan, right? They are present in the cytoplasm only for a limited period of time. After that, usually they undergo degradation, right? Especially in prokaryotic system, in bacteria, usually they have very fraction of seconds or minutes. So usually the average time is around 1 to 3 minutes in bacterial system. At the same time in eukaryote, it may range little more, from several minutes to 7 days. So this is an important feature, that mRNA molecules are synthesized in the system and they will be used for translation and immediately will be degraded, so that the system could save the energy which is required for the translation if the mRNA is subjected for unwanted translation. And again, these are other features. Usually these mRNA molecules are synthesized, all of these things you may be familiar, synthesized in the nucleus through transcription process and will be transported to the cytoplasm in case of eukaryotes. And before the transportation, this mRNA molecule will be subjected for some kind of processing. Already we have discussed in some other video that the RNA will undergo extensive processing at both the ends, like 5' capping and polyadenylation at the 3' end, and moreover there will be some trimming process, some cutting and rejoining called a splicing mechanism to remove the non-coding segments. All those things occurs in eukaryotic system. And this will shows you a representation of the mRNA in prokaryotes and eukaryotes. You can clearly see there in the above upper part of the image, you can see the prokaryotic mRNA, which is usually polycistronic, in the sense there will be more than one gene encoded in the same mRNA. At the same time, eukaryotes are monocistronic, right? And moreover, you can see the 5' capping and the polyadenylation at both the ends of the mRNA molecule. So in simple term, mRNA is a sequence of nucleotides, and these nucleotides will code for amino acids, right? So how these nucleotide sequences will be utilized, or how this nucleotide sequence is read by the protein synthesizing machinery. And this reading pattern is what we are going to discuss as reading frame. Okay, you can see this is a sequence of codons that runs from a specific start codon to a stop codon. Usually that's what mRNA. There will be a start codon and a stop codon, and within this there will be a number of sequences according to the length of the gene. And this particular structure which consists of a start codon and a stop codon is called a reading frame within a mRNA molecule. And occasionally there will be some more sequences even after the start code, even after the stop codon, and also there is possibility some unusual and or unwanted sequences in the mRNA molecule. It's occasionally may be present. Right. So this genetic code usually or the codons are usually triplet codes, and we will see the discussion later. So these triplet codes are usually commaless or non-overlapping and continuously present in the mRNA molecule. So because of this nature, this continuous non-overlapping or commaless nature or in no punctuation nature, the possibility of reading an mRNA molecule is actually 3. That means since the codon is a triplet, you can either start reading from the first base, or you can start reading from the second base, or from the third base. So in all these different readings, you will get different informations. Right? This image will give you that idea. If it starts from the first U, the codon will be UUC, UCG, GAC, etc. like that. If the reading starts from the second base, that means the second uracil, then the reading frame or the genetic code, the first code will be UCU, then GGG, ACC, like that. So the first reading frame and second reading frame are entirely different, and the information encoded by that codons also will be drastically different, right? So again, this image will give you a much clearer idea about the reading frame. You can see there the UCA is there, then after that you can see AUG is there. So in that reading frame 1, reading frame 2 and reading frame 3, usually those reading frame which actually the start codon is correctly positioned, or there is a start codon in the beginning, will be the proper reading frame. So out of the 3, only one will be giving you the start codon at correct position or at the beginning, and that will be the actual reading frame. Okay. The next one is open reading frame. Usually when you have a start codon, this start codon will cause the beginning of translation. We know that. So the start codon, with the presence of a start codon, the mRNA can code for a protein until there is a stop codon, right? So usually within the mRNA molecule or within the DNA molecule, you can find stop codons usually in between. That means usually by around 20 to 30 codons, in between 20 to 30 codons, you can find at least one stop codon, which means that if that part of the mRNA or that part of the DNA is used for translation, you are not going to produce a meaningful amino acid or meaningful polypeptide, right? But if it is a meaningful gene, we know that within the DNA we have only 1.5% of sequences which are having a meaningful information, or which are the genes. So rest of the sequences within the DNA are non-coding. All right, but there also we can see the nucleotides, but these nucleotides will not code for any meaningful protein. This is because even if that part of the DNA is transcribed into mRNA and subjected for translation, they cannot produce a successful protein, right? A complete protein will not be produced because of the presence of stop codons, usually in between around 20 to 30 codons. All right, so there comes the significance of this one, that means open reading frame. Usually a gene will have an open reading frame. Okay, it's actually a DNA stretch which contains around 50 to 100 codons without a stop codon in between. Right, so an open reading frame is a DNA sequence which contains around 50 to 100 codons that begins with a start codon and usually ends with a stop codon. Right, so that stretch of DNA, or even otherwise you can say that stretch of mRNA, is called the open reading frame. Okay, so these ORFs will be translated successfully to produce functional proteins. You can see the representation in the image. So now we are moving into the discussion of a genetic code. So a genetic code is the particular nucleotide sequence of the mRNA which carry the instruction for a specific amino acid. That's the definition. So this is the sequence of mRNA which provide an instruction for a specific amino acid. Right. But we know that actually the information for an amino acid will be present in the DNA. That is what we call the gene. So this gene or the sequence in the DNA actually codes for amino acid. But we know that this DNA is not directly involved in translation. But instead this DNA is converted into mRNA. Or for a decoding process we use this mRNA as an intermediary molecule. Okay. And so there is a rewriting process, right? We transcribe actually. That's the meaning. There is a kind of rewriting. Already the informations are written in the DNA molecule, and these informations are rewritten, right? Written again in another format or in another language, right? So there we use the nucleotide sequences as the alphabets and codon as the words, right? So the alphabets are the nucleotide bases. That means the adenine, guanine, what we usually represents that. So these are the alphabets, and the codons are the genetic code is the words. So each word will be coding for, or each word is a meaningful word, in the sense each codon will code for a specific amino acid. Right. So that's it. The alphabets in the mRNA are denoted as the genetic code or codon, which will specify the alphabets of the polypeptide sequences. Okay. And this idea was actually evidenced or experimentally proved in 1961 by Marshall Nirenberg and Heinrich Mattei. And their experiment was also greatly supported by Hargovin Khurana, who actually helped them to synthesize artificial nucleotides, right? So what are the features of genetic code? So this we will discuss.